This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 439, recorded on April 28th, 2017. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me here in a nice, sunny New York City, Dickson, Despommier.
Hello, Vincent.
It was like raining all week, right?
It's the best day of the year so far, that's for sure.
It was just foggy and raining, and now it's is
remarkably beautiful.
What is the temperature here? New York, 28, which is, according to Kathy Spindler, 82 degrees Fahrenheit.
That's right.
I'm going to edit that out because I don't want to be heard uttering Fahrenheit. Wow, 28 Celsius and sunny. How is it behind me?
It's marvelous. It's absolutely marvelous. I've been at a brief moment for the day, and all the flowers are in bloom. Everybody's dressed in summer garb. The city feels upbeat. It's just a beautiful place to be when the weather's right.
All right. Joining us today from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Is it probably 100 degrees there, right?
No, it is 90.
Wow,
close. You were close.
How much is that? How much is that in Celsius?
It is 32.22222 Celsius.
Nice.
Right.
I read about this is part of my pick. I read about a guy in Google of whom it is said that his PIN number is the last four digits of pi.
That's pretty funny.
That is good.
Google guys have a pretty weird sense of humor.
The last four digits, I mean...
There is no last four digits. It goes on forever. So that means... What are we talking about here?
You pick some way down the line, right? Also joining...
wait a minute. It's sunny. Did I say it's sunny?
90 degrees.
Yeah, it's sunny. I mean, it could be 90 degrees and awful. It's 90 degrees and sunny.
Could be.
It's true.
Okay. Let's go around the country. Joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hello, Kathy.
So, Vincent, it's 61 degrees Fahrenheit here.
Oh, it must be 16.
Know what that means?
16 Celsius.
Yep, you got it.
Boy, I'm just calculating like crazy.
You're so much colder than we are. Wow.
Yeah, it's strange. It's partly cloudy, but
we were 60-ish earlier this week, right?
Yeah. Mm-hmm. So, Kathy, have the tulips blossomed in New Holland yet?
I don't know about in Holland.
No, New Holland.
What's New Holland?
New Holland, Michigan.
Holland, Michigan.
Oh, okay.
I don't know about New Holland, Michigan.
Holland,
the town where Hope College is Holland, Michigan.
Okay.
Is that like Harry Ginsberg playing football for University of Michigan?
For Michigan. It is kind of.
Stop. It
Where
hurts.
did Norwalk virus come from?
But to be exact, the tulips are also blooming here in Ann Arbor.
All right.
Yes.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's sunny and humid and 81 Fahrenheit, so I'm not on one of the palindromic ones. But
You're close. You're pretty close.
Yeah, it's not bad. And yeah, it was rainy and cloudy all week.
Yes.
Yeah, it's been a foggy, rainy week, but now, appropriately on Friday, it's sunny. All right, we have a guest joining us today in studio. In fact, he just arrived as we were introducing each other. Perfect timing. He is an investigator of the Howard Hughes Medical Institute. He's also a professor at the Laboratory of Retrovirology at Rockefeller University, right here in New York City. Unfortunately, on the other side of town and all the way downtown. And Paul Bieniasz, welcome to TWiV.
Well, thank you very much for having me. I'm delighted to be here. And before we get going, I have to tell you a little anecdote that might amuse you. So I listen to a number of podcasts, and sometimes I listen to them in bed when I'm just about to fall asleep. And sometimes I fall asleep with the podcast still playing. And sometimes that podcast is TWiV.
Yeah.
And when that happens, I end up having a dream. I dream that I'm a guest on TWiV.
Wow.
And because I'm, you're obviously
wake up. You're late for work. You've got to wake up.
Not going to stop to let me speak. In my dream, I'm trying to get a word in edgeways, and you guys just won't stop.
Doesn't work. That's not going to happen here.
So in a very real sense, today a dream has come true.
That's great. That's funny. A dream come true. Well, a number of people tell us that they fall asleep listening to our podcasts.
Or pipetting.
The hard part is when they say, I listen to it to fall asleep instead of while falling asleep.
Yes.
Not a problem.
Not that the content is anyway soporific.
Of course not.
Soporific.
Soporific, that's right.
Yeah, we are not laconic nor soporific.
It's sophomoric, but it's not soporific.
What other podcasts do you listen to?
Well, I kind of flit around a little bit. One of my favorites at the moment is Sam Harris's podcast.
Mm-hmm.
I don't know if you're familiar with that one. I occasionally listen to Joe Rogan if he has an interesting guest. Some of the podcasts I listen to are political.
Yep.
Sometimes not aligned with my own views, just so I jump around a bit.
Huh. Sort of like a transposon.
Yes. TWiV is a regular.
TWiV is not just a podcast,
No.
Dickson, you know.
No. It's an open reading frame.
All right, I want to tell you about the ASM grant writing course. Learn the best practices in grant writing during the ASM grant writing online course. Participate in the course to receive an overview of the funding landscape, learn what makes grants successful, and receive personalized feedback on your grant. This is a 7-part series taking place in the fall of 2017. Dixon, maybe I should take this series, given my recent lack of success. Fall of 2017. Registration deadline July 15th. Learn more at bit.ly/ASMGWOC17. There'll be a link in the show notes.
Now, of course, everybody eventually gets personal feedback on their grant, but this will hopefully be more useful than just, you're not funded.
Exactly.
You know, I've learned that no matter how many years you've written grants and how successful you've been, there's going to become a time when you're not going to get it, and therefore all the things you thought you were doing correctly simply don't work.
That.
So I don't know.
Yeah.
You know, you can't teach that because
I had 6 in a row before that happened, so I was kind of happy with that, but I never knew why I got it.
Survivorship bias.
I thought I deserved it, but when I didn't
keeping
get
doing
it,
what I was doing
before.
Yeah.
Yeah. Unfortunately, we have sad news. I heard this morning from Carolyn Coyne that Julie Youngner died last night.
Really is sad news.
And he was in his 90s. You know Julie Youngner, Paul?
I don't know him. I actually know of him from your podcast.
Okay. Yeah, he was on TWiV 373 when I went out to Pitt and did a podcast, which was a really nice podcast. He was just so personable. And he, of course, had a long and distinguished virology career, perhaps best known for working with Jonas Salk when he was at Pitt on the inactivated polio vaccine, but also did lots of other things in his career, and we talked about those on the TWiV. So my last memory of Julie, we went to dinner, and we were leaving, and he gave me a big hug. So that's the last memory of Julie. So
That's
thank
a
you.
nice memory.
Check out the podcast if you haven't listened to it, because it's got video, and he's great. And I'm glad I was able to record him talking about his career, right?
Absolutely.
For an hour and a half, not just 10 minutes. You can get your hour and a half of recorded history. Similar fashion, Philip Marcus at UConn, who came up. So I recorded a podcast with him, and then 2 years later he died. This is not a trend, Paul, okay?
Oh, my goodness.
But
We have a lot of guests who've
I'm
been
gonna
on the
leave
show
now.
who are
Lots.
still
But,
alive and well.
That's right. Some of them die on the show.
Philip Marcus was mentioned by Andy Ball.
No, I mean in another way.
Philip Marcus was mentioned by Andy Ball when he talked about his classic paper mapping of VSV transcription. Remember, he did talk about Philip Marcus being part of that group and giving him good ideas. So anyway, sorry to hear it.
It's the TWiV bump. It's not the TWiV bump off.
Excellent.
That's good. All right, just 3 follows up from the last TWiV. Ajinkya writes, Dickson was pretty awesome in TWiV 438. No, really, he did.
That's what it says.
So Ajinkya is anticipating me saying, no, you weren't. And okay, thank you.
Well, no, I think maybe saying he really did go and shake hands with everybody at the Science March.
Did you?
Yeah.
Like he was
tried.
manically promising to do.
I just stood still. Everybody else came by me. That's what happened.
How many was that exactly?
How many people were there?
A few tens of thousands. You must have had a sore hand by the end.
Well, I did this and then said times 10,000.
Okay.
Well, he said he was going to then rub all their DNA into his hands. He's going to transfect himself with all
That's right. That's right.
Are you going to get lateral gene transfers?
I don't know.
Do some virome analysis.
I have no idea what I was talking about, actually, but I had a great feeling during the march.
Nadia writes, "Hi, all in TWiV land. Last Friday, I was glad to get to meet TWiV co-host Kathy Spindler for the second time when she gave a seminar and workshop during the Yale Science Journalism Symposium on communicating science. During one of the workshops, Kathy gave us exercises to help us practice communicating science. For one of the exercises, we had to describe our favorite protein and what it does in the first person. I chose the protein I am studying, which is an abnormal fusion protein in acute megakaryoblast leukemia. So naturally, I acted like a villain. Kathy made it fun, and I learned a lot about how to describe science in an accessible and clear way. Thanks, Kathy." An issue that arose during the symposium is that while we all can agree that science communication is important, some worry about the impact of being heavily involved in communicating science in the popular media will have on their career. Kathy made the point that I would like to echo, that active scientific communicators should be rewarded and not be penalized. To that, I would add that the success of TWiV and the existence of a TWiV bump is evidence of how valuable communicating science is and how everyone should and can do it. Apart from communicating science, the TWiV co-hosts have created a community, the community of TWiV listeners from near and far. Scientists to science lovers is a powerful force for science. Thank you all, and keep doing what you're doing. Yep. Well, I would say that maybe being heavily involved in communicating science would be too much, especially for a young investor. You can certainly be involved in communicating science, but you have to balance it, obviously, right?
That's exactly what I said. It was somebody who was starting out in his career, and I said, well, if you took things on and did things at the level that Vincent Racaniello is doing yeah, it could hurt your career, but there are things you can do. And I think that it should be something that should be rewarded and noted in annual reports. And I think that it will improve networking and could improve the research science that this person could do, for example, by involving himself with other people. So,
yeah, I don't see any rewards here, Dickson.
No, I don't either. I once knew someone at another university, obviously, who every time they discovered something, it didn't matter how trivial or how important it was, they called the New York Times and said, I've got a breakthrough. And they got the New York Times science reporter on the telephone, and after a while it became tiresome because they realized that he was calling like every other day almost. And and he became known as a person who just wanted to be in the spotlight.
You know, David Baltimore used to do that, but he was restrained. Okay.
By who? Mrs. Baltimore?
No, he didn't call. He knew Lawrence K. Altman, who was he was an MD. And I remember when we got an infectious polio clone, it was accepted in science, and he called.
Oh, that's
Altman,
important stuff.
And he said, actually, it was Harold Schmeck, who was another science writer for the Times. But he called them, and the guy wrote an article about it.
But Vincent, that's an important finding. This was everyday stuff.
No, you have to moderate. If you have a connection, you don't want to overuse it,
that's
right?
That's exactly right.
So I don't have any connections.
Yeah, I think, everyone has a certain amount of bandwidth, and you have to divide your efforts accordingly. And all of us might have slightly different roles. I mean, if bench science is your first love, you're not going to have a lot of time for communication, and vice versa.
So when the press calls you, do you accept mostly?
Oh, yes.
You do. Yeah, that's right. That's the right idea.
But that's typically not every day.
Well, look,
we'll try to call more often now.
Okay.
I don't want to prolong this, but early in my career, I still love bench science, so that's all I did, right? I had a relatively good-sized lab.
That's true.
But at some point, I got interested in communicating, and I decided that it made a bigger impact than doing experiments, so I downsized my lab, and eventually, in a few years, it'll be gone, and I'll just do this. But everyone has to find their own level, but you'll do something. You could teach once a semester in a high school. That would be great.
Absolutely.
Right? Something like that, because high school kids love it when scientists go and talk to them and bring them plaque assay plates, unused, of course.
Yep.
Horses for courses.
What's that?
Horses for courses.
What does that mean?
Well,
To each his own.
Different kinds of horse do better on different kinds
courses.
of courses.
I'm thinking of courses and like classes, but he's talking about race courses.
You have race horses.
Got it.
Sprint or hurdle.
That's right. That's right.
All right.
That's right.
Coming back to last week's TWiV that you guys did, it was at the same time as I was doing this symposium. And when I listened to the TWiV, it was really fun to hear how many of the same themes came out and the same kinds of concepts and advice and things like that. Yeah. So I thought it was just a really interesting confluence of events last Friday.
Sure.
One of the things that we had an audience question last week. Someone asked, should scientists or non-scientists communicate? And Alan, of course, said both, and that's correct. And the reason why I bring that up is this past week there's been a lot of Twitter chatter about science communication, obviously because of the Science March. And I noticed today and yesterday a number of people are saying, Bill Nye and Neil deGrasse Tyson, they're kind of getting old. Can't we get some fresh, young communicators and maybe some women? Mm-hmm. And so I tweeted to them both, we have this thing called Microbe TV. We're not Bill Nye or Neil deGrasse Tyson, but we do have women. And I think, we can do a pretty good job of communicating our life science anyway. So one thing I learned is to start answering people like that who say things like that.
I don't think there's a shortage of young people who are interested in this area. And if the people who are the best at it will make a name for themselves, and they'll be the next generation of science communicators.
Yeah.
When I talk to young people about how to conduct science or what it was like to be a performing scientist in the laboratory, so to speak, a stand-up parasitologist is what I used to be known as. They're amazed with the amount of routine work that goes into producing a result. All of the maintenance of things that come together on a particular day, and finally you get to do the experiment, and then, of course, it's the excitement of the find. And the amount of work that goes into stuff like this is, they don't look at, they think that you just sit there and discover stuff in your head and then say, or something like that. But it's obviously fascinating work, as we found out when we started to talk about how pleasant it was to talk about plaque forming.
Well, that's the point, is that if we try and give a little bit of the behind the scenes, right, Dickson? Yeah, that's the wall of polio, Paul. I don't know if you've ever seen that, eh?
I've seen it on photographs, never in
not
real
live.
Yeah,
it's cool.
Is it bigger in
your
life than it was in the photograph?
Will be posted.
That's right.
Did your biosafety committee know about this?
Yeah, they've been
Everything
in here.
is completely inactivated.
Yeah, they've been in here. Paul, where are you from?
So I'm, as you can detect from my lack of accent, I'm from the United Kingdom, where the language that was invented.
We resemble that remark.
I moved around a lot as a child, actually, but my hometown is in a very quiet rural area called Lincolnshire. Mm-hmm. Famous for not very much, except that the high school I attended actually was where Sir Isaac Newton learned his trade. So, a few hundred years before me, of course. Yeah. So yes, my home is the middle of England, but I haven't lived in England for almost 20 years now.
So I'm looking at it. It's kind of...
Is that by choice?
It's east,
yes, it is.
East of Nottingham, right?
That's right.
Leicester.
Yes. Robin Hood country.
It's not quite Yorkshire, right? It's south.
No, little south of Yorkshire.
I'm trying to... I know, the only person I know with a Yorkshire accent is Jeff Almond. Did you know him?
I don't know him personally.
All right, so he had Yorkshire. But you don't have a Yorkshire accent, right?
No. So because my father was in the forces, we moved around an awful lot.
Right.
If anyone has a generic accent, it's me.
It's a generic accent?
Yeah. Or no accent.
You have an accent to it. What is a generic American accent? None.
Most of the radio
announcers. It's what you hear on most news shows and radio shows. There's standard American English.
Alan's got a perfect
by the way,
accent.
Mark Twain once commented that the King's English is not the King's. It's a joint stock company, and Americans own most of the shares.
That's great.
So
that was a hostile takeover.
It certainly was.
Mark Twain was great. Mark Twain.
You're from the middle of England. Why did you Brexit?
But
I think, like many people in my situation, one comes to the US at some point in one's education. In my case, it was at the postdoc level. And I came to the US sort of thinking I would probably return to the UK, but not being married to the idea, and just found the opportunities here were just so much better. And that's really why I'm here now.
Where did you get your PhD in the UK?
So I studied with Jonathan Weber initially, and then Myra McClure, whose name you'll recognize from the
XMRV.
That's right. Yes.
In fact, I interviewed her a couple of years ago.
Yes, I remember that.
Yeah.
So I worked initially on trying to identify the then unknown HIV co-receptors, and then moved on to a project with Myra, which was on the foamy viruses, sort of the underrepresented minorities of the retrovirus field. But a very, very good learning area, not so competitive as many areas of virology.
What got you interested in science to begin with?
That's hard to say. It came to me rather late, actually, and gradually. So I never really wanted to be a scientist until I actually worked in a lab as an undergraduate. So as a kid, my family has a long history of being associated with the Royal Air Force. And so I really wanted to be a pilot, strap a jet fighter to my back and...
Damn right.
But it turns out they won't let you do that if you don't have perfect eyesight, which I don't. So I had to
Alan's
rethink.
a pilot, by the way, in case you didn't.
Right.
But not, I don't fly fighter jets.
No.
Neither do I, sadly. So then I thought I might want to be an engineer, and so I studied maths and physics and chemistry, and found that I liked chemistry much more than maths and physics. And then started to get a hankering for biology. So as an undergraduate, I studied biochemistry at the University of Bath. And during that time, you got to spend some summers working in labs away from the university. And it was one of those stints. One summer working in a laboratory in a pharmaceutical company, at that time called Sandoz, now called Novartis.
Sandoz, of course.
I had a wonderful mentor, gave me a lot of freedom, even at that young age, and that's when the bug bit, and I knew this was what I was going to be doing for the rest of my life.
I've been to Bath. They have the hot springs there. Right?
That's right.
That's where the Romans were the first to take advantage of.
I was a meeting nearby in Cirencester.
Yep.
You know that town?
I do, yes.
There's a university there, actually. In Cirencester.
Yes, that's probably one of the newer universities.
Yeah, there was a meeting there.
And there's a Rock of Ages someplace in the area, too, as I recall.
The Rock of Ages.
Yeah, there's an old song about the Rock of Ages. And as my host was telling me on my way to Bristol with him, he says, see that rock over there? That's the Rock of Ages.
Oh. Well, I was there for 4 years and I missed it.
Maybe it crumbled in the meantime. Who knows?
So you came to do a postdoc at Duke, right?
Yes, with Brian Cullen. And that was sort of the movement into the big leagues. And Brian had a bit of a tough reputation at that time, but I actually found his lab to be a wonderful place to work. He's been a very supportive mentor to me, both while I was in his lab and since, actually.
What did you work on?
So right around the time I went there, the co-receptors, the HIV co-receptors had been discovered. And so we were mapping bits of the co-receptors that were important for their function, how they interacted with the HIV-1 envelope, how different strains interact differently with different receptors. You'll know that there are two different co-receptors, for example, and how that transition is made. Then after that, I tried to identify cofactors of the HIV transcription factor, TAT. It was known that TAT was a very potent transcriptional activator in human cells, but didn't work at all in mouse cells. And so we, based on some work that Brian had done previously, we strongly suspected there was a human-mouse difference that could be exploited to try and identify cofactors. And while we were right about that, it turned out that somebody else discovered the said cofactor. But I was very well positioned to jump into that field and make some contributions and find that the reason why TAT doesn't work in murine cells is due to a single amino acid difference between the human and murine proteins.
Wow.
That's
It always needs to be confirmed, though, isn't it, in the literature?
Sure. Reproducible results are good. It's just best if somebody reproduces them after you.
That's right.
Absolutely.
That's right.
Absolutely.
So then, after your postdoc, did you come to New York at that point?
That's right. So while I was a postdoc at Duke, the person who is now my wife, Theodora Hadzioannou, she was doing her graduate studies in France, and we actually met at the Cold Spring Harbor retroviruses meeting.
How romantic!
It's true. It's true.
I met my wife at a scientific conference. It's a great place.
It was extremely romantic, and you'll be surprised how often these things happen.
I wouldn't. I used to run a Gordon conference.
Anyway, for a while, my then girlfriend, future wife, had a transatlantic relationship, and came to the decision that we wanted to be in the same city. That immediately restricts the places you're going to look for work to a few places. And so Theodora got a postdoc position with Steve Goff right next door to where we're sitting now. And I found a position at the Air and Diamond AIDS Research Center towards the end of 1999. And so that's when we both moved to New York at the same time.
I remember when Theodora was here.
Do you?
Yeah, for sure. Quite a few people have passed through the Goff laboratory, and I know that Theodora, I remember her very well, and also Alice Telesnitsky, who's at Michigan, was here for a while, and many others as well. But then she left, and I wasn't quite sure where did she go after her postdoc, right to the Air and Diamond, or?
Actually, yes, Theodora came to work with me. We found that, not to go into too much details about how young couples get to know each other. We were, living together for the first time. We found many of our conversations at home were about our various science projects. We were basically interested in the same thing. And so we thought, well, let's kill two birds with one stone and work together. And so Theodora actually joined my lab as a postdoc. And we've sort of been working together as collaborators ever since. So she had a separate lab at Air and Diamond, and my lab was sort of across the hall from hers. And I have some projects, she has some projects, and some projects we share. And we've been working together for many years now as a team in that way.
We'll have to have her on at some point. Does she fall asleep to TWiV? Do you know?
She's usually fast asleep by
Maybe I shouldn't have asked that, right?
That's right.
I was very happy to invite her to ASV a couple of years ago.
Yes, she enjoyed it very much.
Retrovirologists don't come often because there's a concurrent Cold Spring Harbor meeting, which is not too far off from ASV, right? And so a lot of people don't go.
I'm going to be there this year. I'm coming to ASV.
ASV, great.
Yeah.
Anyway,
excellent.
So she loved it, and she told me when I saw her a few weeks ago at Rock that she likes to go back, and she tells her lab to go because she really enjoyed the meeting. Right. So that's great. Yeah, really happy about that. Now,
pardon my naivete, but what's the relationship between, or is there one, between the Air and Diamond Institute and Rockefeller?
Yep, that's next. Tell us.
Okay.
Right. So the Air and Diamond AIDS Research Center is actually a phenomenal little center. It's academically affiliated with Rockefeller. It's administratively and financially, they're a completely separate organization. So in a sense, working there, one had the some of the advantages and disadvantages of both types of work environment, small institute and while being affiliated with Rockefeller University. Actually, very recently, I've moved my laboratory from the Air and Diamond to the Rockefeller campus. And while the relationship between the Air and Diamond and the Rockefeller still stands, I am now fully a Rockefeller associated rather than being associated with the Air and Diamond. Although I was there for 16 years, and they were great years. Wow. I really enjoyed my time there.
Which building is your laboratory in?
My laboratory now is in the Rockefeller Research Building, second floor, overlooking the East River.
Oh, yes.
It's a new one, right?
Yes.
Nice.
Yeah.
Yes.
So, Paul, it looks like being associated with the Air and Diamond Center, you had you had to go to some gala events that included, for instance, Bill Clinton, and there's a nice photo of you and Bill Clinton and other people, David Baltimore, for example.
Yes, so there is a photo that exists that it only came about because Bill Clinton was late showing up to give his speech. This was a fundraising event, and my only job in this fundraising event was to introduce David Baltimore, and David Baltimore was going to introduce Bill Clinton, and Lucy Liu was also there as the MC, and Magic Johnson, who also is well known to David, was there too. And so everyone's sitting at their dinner table having dinner and so on. And I was supposed to get up 5 minutes before and go and introduce David Baltimore. It just so happened, as I was waiting to go on to introduce David Baltimore, Bill Clinton arrived. Okay, so in one room at one time, there was Lucy Liu, Magic Johnson, Bill Clinton, David Baltimore, David Ho, myself. And there's a photo that captures that.
Outstanding.
That's cool.
Outstanding.
Where'd you find that, Kathy?
Well, you just do Google image search for Paul Bieniasz, and it's toward the bottom of the first full page of images.
You could do Bieniasz and Clinton together, right? Well, that's pretty cool.
So my other question is, what is the origin of your last name?
That's Polish. My grandfather was Polish. He left Poland at the outbreak of the Second World War.
Smart.
You say smart, but he got on a ship that he thought was coming to the United States, but actually ended up in Liverpool, England. So I'm basically got here 2 generations too late.
So I found one. I see the picture you're talking about, but there's also one of Ho, you, Theodora, Lucy Liu, Steve Goff, and Beatrice Hahn.
Yeah.
Which is a nice one as well.
That's also good.
Wow, these are cool. Yeah, there's David, and Magic is towering.
Yes.
Above
and it takes quite some height to tower over Bill Clinton.
Well, that's right. You're taller than Bill, according to this picture.
We're about the same, I think.
About the same.
I remember.
Maybe it's parallax.
He has a way of making himself appear taller, I think.
I think being a former president of the United States may have that effect on people.
Yeah, why not? You might as well. You got elected. That's no easy thing. There have only been 44 presidents. Oh, did I miss one?
No,
nope.
You didn't.
All right. So, Paul, we got an email a couple of weeks ago from Daniel Blanco Melo.
Yes, my former student.
He wrote, I'm a recent graduate from Rockefeller University from Paul Bieniasz's lab, and our paleovirology work was just published today in eLife attached file. In this work, we resurrected the envelope protein of a primate endogenous retrovirus and found that a copy of this envelope gene, present in the genome of humans, gorillas, and orangutans, was most likely co-opted to combat similar retroviruses in the distant past through a mechanism resembling receptor interference. I thought that you might be interested in this publication. I wanted to share it with you. I think it's funny that, you probably wanted to get it on TWiV, right?
Well,
sure.
I had no idea he had done that.
So
perhaps he should have called the New York Times.
Exactly, right.
Well,
unfortunately, no one's there anymore. So.
This is what I wanted to discuss with you today. And but this kind of study, the Times would love, because people are always interested in very old viruses and resurrecting them and so forth. So that would be something Carl Zimmer would write about, I'm sure.
Sure.
Right. Well, I'm fairly sure that both the university and eLife did a press release.
Yeah.
And, it just depends,
of course,
who sees it, what else happens to be on the agenda that day.
So Daniel was your PhD student?
He was, yeah.
Now he's a postdoc at Mount Sinai, right? Who is he working with?
He's working with Ben Tenover now.
Yep.
Yeah.
Ben's been on TWiV.
Yep.
All right, Daniel, so here's your 15 minutes. Maybe you can get more in the future. You never know.
You don't.
Now,
we give people more than 15 minutes.
Yes, we do. In fact, we give them an hour and a half to 2 hours, right?
Yes.
Although we know Kathy has to leave at 4:30 today. So you can alert me if I forget a few minutes before, so we can.
So we probably shouldn't spend the first 40 minutes of the show leading up to talking about the. Oh, never mind.
It's okay. It's okay.
That's all right. I enjoyed it.
Yeah, that's fun. It's fun. And, what do they say? If you want to learn about science, learn about the scientists.
Learn about the scientists, absolutely. Right? No, I was not criticizing that, just joking.
I'm very sensitive to criticism. Right, Dickson?
Exactly.
All right.
Be nice to Vincent.
No, it's okay. You don't have to be nice to me. Be nice to Dickson. This is an eLife paper, Co-option of an Endogenous Retrovirus Envelope for Host Defense in Hominid Ancestors. And there's Daniel and you are the first and last authors. And then Robert Gifford, who's over in Glasgow.
Yeah.
And we did his paper a couple of years ago where an interesting story about Plasmodium and a retrovirus. Do you remember that story?
Oh, yes. He found that, in fact, you have to remind me.
Put you on the spot, yeah, because I don't remember. The origin of a
mammalian, an avian, what's now an avian retrovirus, had its origin in mammals and was transferred there during experiments using the malaria parasite.
That's right.
And it appears to have escaped into nature and.
Wow.
Yes.
Yeah, that was
not
an
great.
Interesting story. So Robert, and I met him at ASV.
Wow, have we done that? We haven't done that paper.
Yes,
right?
we did.
Yeah, we
We did.
Rich Condit, you had some communication with the first author.
Oh!
Forgot her name.
One.
Yeah.
Yes, that one. Okay.
Just a little backstory. I can remember when Rob was trying to convince me that this is what happened and given rise to spleen necrosis virus. And he was a little worried about sort of publicly presenting this. And I said to him, yes, you better be right about this. If you say this and you're not right, then
they'll remember
it'll be
that
difficult.
too.
Yeah.
So this idea of looking at endogenous retroviruses, you've kind of worked on this over the years, right? You've published a few other. I mean, you do many other things which we're not going to unfortunately be able to talk about, but you've recovered an ancient retrovirus. So tell us. Let's start by talking about what is an endogenous retrovirus.
Well, basically, an endogenous retrovirus is a virus that has infected a cell that either is or would become part of the germline. Mm-hmm. And in so doing, provided that doesn't cause too much of a fitness cost to the host.
Like killing it.
Right. Or at least making it not unable to reproduce.
Yeah.
Right.
Then retroviruses can be inherited in much the same way as Mendelian genes. Mm-hmm. And again, provided the fitness effects aren't too deleterious, over time, through drift or even through positive selection, those proviruses can become fixed in the genome of that species as the species passes through bottlenecks, particularly. And over time, that those proviruses accumulate, and there are many, many, many different types of proviruses in the genomes of basically all modern organisms. Mm-hmm. Humans, for example, about 8% of our DNA is viruses that used to replicate in the same way that normal retroviruses replicate, but some of them have found their way into the germline and have accumulated there. And it constitutes a fairly rich fossil record of retroviruses that have infected our ancestors over millions to tens of millions of years. There's some problems going back further than that, because once you get into the hundreds of millions of year time frame, then things start to degrade through neutral mutation that they become more difficult to recognize. But in the millions to tens of millions of years range, there's quite a nice record of retroviruses that have infected our ancestors.
How can you tell how old they are?
There are two dating methods that we use primarily, none of which are particularly precise. The first is quite simple, is its occurrence in a particular range of species at a specified location in the genome. So when a retrovirus integrates into the genome of its host, it integrates at a specific site, okay? Mm-hmm. And it will never move from that site. So say, for example, a retrovirus infected or became integrated into the germline of the primate that's the ancestor of all modern chimpanzees and all modern humans, you'll find that in every human, pretty much, in every chimpanzee, pretty much, unless they've happened to have deleted that locus, will have that provirus in that precise spot. If it was in the ancestor of modern humans and chimpanzees, then that provirus will not be present in gorillas, orangutans, gibbons, monkeys, and anything that branched off the phylogenetic tree prior to that insertion. And so you can get a rough estimate of the integration time based on the occurrence in individual species.
So that dating, you use the known dating of the species.
Exactly. Based on independently generated dates of when species diverge from each other.
And of course, these are species that are on Earth at the moment.
That's correct.
And we assume, but we know that how they were related to one another, but there are many others that have died out and we can't possibly study. And
so greater than 90% of all species that have ever existed are extinct. The same's probably true of viruses too. We actually don't know that number, but I would suspect it's probably even more than 90%.
How closely related are all these proviruses of a single kind that originated, like, say, 10 million years ago? How faithful is the reproduction of that genomic sequence that allows you to say what you're saying?
Okay, so unless there's selection pressure acting on that proviral insertion, and it can be positive, rarely that provirus might be co-opted for some function, as we suspect is the case in the paper we're going to talk about today. Or there could be negative selection if the provirus is in an important gene, for example. It could be mutated to try to get rid of it. Absent no selection pressures, the proviruses mutate pretty much at the neutral mutation rate of the host. So in humans, about 2 times 10 to the minus 9 substitutions per nucleotide per year. Okay, and so that actually provides the basis for the second dating method we use. And so if you make the reasonable assumption that when a provirus integrates into the host cell DNA, the sequences that are repeated at its end, 2 so-called long terminal repeats, if you make the assumption those sequences are identical at the moment of insertion, and then you count the number of differences in those 2 LTRs, provided they survive to modern times, that gives you a second independent estimate of age. And give or take, I mean, these are not precise dating methods.
Right.
Give or take, those methods generally are in broad agreement with each other.
Plus or minus a few million years.
Right.
But who's counting?
But when you're dealing with a slowly evolving species like hominids, that's not a big problem.
Right.
The second method, the sequence drift method, makes the assumption, does it not, that once integrated, that provirus is inactive or becomes inactivated fairly shortly thereafter. Is that correct?
It doesn't make exactly that assumption, but it does make the assumption that the provirus is not placing so no selective forces are acting on that on that provirus. If it was expressed or causing some disturbance to the host, then you would expect some selection pressure to be acting.
Yeah, because I'm thinking that if you got a number from the sequence drift that was a lot smaller than the number that you got from the species association, would that not imply some sort of positive selection, and maybe that for a period of time this thing was active?
It would, to the benefit of the host.
To the benefit of the host, okay.
Right, because the host preserved that sequence. So this is integrated retroviral DNA, which is then going to be replicated by the host. And so if the host is preserving those sequences and replicating them, then there's some selective pressure and I guess conversely, if you saw a much faster mutation in them, there might have been some negative selection.
There are some anomalies. For example, you can get homologous recombination between proviruses in different loci. And so then you can get deviations from that invalidate that dating method.
Now, there are 8% of our genome roughly is endogenous retrovirus, right? And many of them, or some of them, have been shown to be exapted or used by the host for functions like the syncytin protein. Right. And you also mentioned in your paper transcriptional regulation. We did the Nels-Elde paper where they show the promoters for interferon responses are taken from LTRs. And so do you think that ultimately all of this 8% will be shown to have some role, or is a lot of it just sitting there? And
No. I think a very small minority of it is actually exapted.
Okay.
Yeah. That junk DNA is a real thing, right?
It's a real thing.
Yeah. So
Richard Dawkins is right.
Provide
Brits also think alike, by the way, so we should be careful here.
Provided a piece of DNA does not place a large negative selection pressure on its host, the pressure to remove it will be minimal.
Really?
And yeah.
When you think of the metabolic energy, though, it takes to make a gene and to replicate the entire genome, and 8%.
But when you're already replicating billions of other bases,
yeah, no, I got it, but 8%.
the margin is very
that's
small.
a big percentage, though. 8 to 10%. If you could get rid of the energy necessary for 10% without a risk,
but to a cell, it might not.
Well, I think maybe that's where the concept.
You are anthropomorphizing. That's the problem.
No. I'm just looking at energy.
No, I mean you make a good point that a small advantage or disadvantage can be evolutionarily significant. Right. But I think probably in the context of replicating cells, this is just not enough of a load to be worth the considerable energy expenditure to remove these, because in removing them, you'd have a considerable risk of removing something you need.
Yeah. So cumulatively, that there is a burden of junk DNA.
Yeah.
But let's say you had a mechanism to get rid of 10 kilobases at a time, right? That would have an absolutely negligible effect on your fitness. Right. If you try to do it in bigger chunks, then you'd rapidly get into trouble.
So isn't it the case that recently, or maybe not so recently, someone has removed all the endogenous retrovirus from porcine cells for use in transplantation?
Certainly not all of them.
Not all?
I'm not familiar with the work, but I think that would be impossible. They may have targeted a few specific loci that encode replication competent proviruses. That's doable.
Yeah.
But taking them all out, that is an engineering feat. That is, I think
the number possible. 30. Someone told me. I think Goff said 30-ish.
Okay.
And I'm wondering if that makes the cell replicate a little better or whatever, right? But I don't think they looked at it. But removing all of them would be hard. Okay. Yeah. So this story, tell us how you came about to do this. What was the backstory here?
So, as you mentioned, over a number of years, we've been interested in resurrecting. I know you don't like that word when we're talking about things that aren't living. Right? I'll stop using it when you stop saying live vaccines.
You didn't. I try.
Touche.
You didn't use it in your title. You said reconstitution of an infectious.
Yes, I know.
That was very good.
We do deliberately avoid the term resurrection these days because it is.
I had a big argument with Carl.
Certainly,
Zimmer
Richard
about that.
Dawkins would totally disagree with that concept.
Well, this paper did come out around Easter.
Yes.
Stop it, all of you. Ooh. Come on.
Anyway, where were we? So. As I said, over a number of years, we've been interested in reconstituting these defective retroviruses for a couple of reasons. One simply is because we can, right? This is something that wasn't even possible until our ability to synthesize big chunks of DNA became readily attainable, and it actually occurred to me. What is it, about 10 years ago, that you might be able to do this simply by synthesizing consensus sequences based on fossils that are present in the viral genome. And so Thierry Heidmann had the same idea about the same time, and we published these papers about HERV-K, which was really the low-hanging fruit because that's a very young, recently extinguished retrovirus.
Dickson, ask him how young it is.
Okay, how young is it?
It passed into oblivion, died.
Yeah. It's no longer useful.
Within the last few 100,000 years.
Oh, that's within the span of human history.
Since humans have been humans.
That's
right.
So I have to ask, as long as we're talking about this, are there, how shall I say, biosafety issues associated with reconstituting a dead retrovirus?
Inactive.
There are, and I should state very clearly what we don't do is synthesize complete proviruses and then transfect cells in a laboratory and then
Right.
What we're doing in these experiments is typically to reconstitute individual viral proteins and then incorporate them into replication defective vector systems. So you'll know that, for example, that you can pseudotype retroviruses with envelope proteins from other viruses. And so what we did in this paper, in fact, was to basically rebuild the envelope protein and put it on a modern retrovirus that is only capable of a single cycle of replication. It's really a retroviral vector. We reconstitute retroviral vectors rather than full-length retroviruses.
So in the case of ERV-K, you never really actually put together the whole thing. You recovered pieces of it and showed that the individual pieces worked as expected.
Right. In the case of ERV-K, we did make a full-length proviral plasmid, and then what we immediately then did was to replace the envelope gene with a selectable marker, an antibiotic resistance gene, and then we complemented everything in our replication assays.
Okay.
So simply being able to do this, to study viruses that aren't around anymore, I thought might be interesting. But the real sort of confluence of this work with my other interests was to try to find out what made these extinct viruses extinct. So a big activity in my lab is to discover and characterize antiviral genes that provide a defense against modern viruses. And of course, if you think about it, if those genes actually do their job and succeed in eliminating a virus, then that virus no longer exists anymore. There's no longer, in principle, potentially no longer a need for that antiretroviral protein, and that antiretroviral protein can then fade from existence, having done its job. Really thinking of the innate immune system as really an adaptive immune system, but adapts over evolutionary time rather than in the lifetime of the host. So I've been interested in trying to understand how these host-virus interactions play out. And so Rob actually was at the Aaron Diamond AIDS Research Center for a time, and he's, as you know, is very interested in describing the evolutionary history of retroviruses, and we thought it'd be a great idea to team up and try to do this in a somewhat systematic way, go through the genome, find old retroviruses, reconstitute them, and use our complementary expertise to try and find out how retroviruses became extinct. And HERV-T is an example of a retrovirus where there's a decent number of fossils. Obviously, for the reconstitution effort to work.
You don't like that term, Dickson?
Not particularly.
You don't like the term fossil?
Fossil, yeah. Okay. I resemble that remark.
Does it have to be a stone if it's a fossil?
They have not mineralized.
These are sort of the oldest stories ever told in DNA sequence.
Right.
I think fossil's okay. I like it.
You guys really got to lighten up, okay? Yeah. Just lighten up.
Who has to lighten up?
It's okay.
I think it's fine.
Okay.
I don't have a problem. Yeah. Sorry, go ahead.
I've heard you guys are all really pedantic.
We are pedantic. Yeah. It's just what we are, right? We happen to be, and a lot of scientists are, and some are not. Are you pedantic?
No, not at all.
Not at all, yeah.
Not at all. I'm constantly using the wrong words. That's fine. I think provided you can make yourself understand.
You invented the language. I mean,
exactly.
You can use it any way you want.
I will not take lectures on the English language from America.
You will not put.
That's great.
You're constantly using words that other people take exception to. They aren't necessarily wrong.
Right. Yes. I just want to make it clear what he said because it was kind of lost in the noise. I will not take lessons from an American on the English language.
Yes.
I think that's fine.
Okay, so these DNA elements that are analogous to fossils
Perfect. There must be an acronym for this somewhere.
For this particular retrovirus, HERV-T, there are plenty of them in the human genome and the genomes of other primates. And so when you're considering doing a reconstitution project of this type, then that really helps to your success, because basically what you're doing is taking those fossils and then trying to reconstitute an evolutionary history of how they came about. In a sense, turn back time to find out what the sequence was tens of millions of years ago that gave rise to all these fossil-like elements in modern DNA.
Of course, you could do this because we have so many genome sequences, right?
Right.
So did you just mine them, or did you do any new sequencing, or you didn't have to do that?
We did no new sequencing. So the innovation that Rob, with the help of Daniel, made was sort of an iterative blast searching algorithm. So they use a catalog of modern retroviruses to blast search primate genome sequences, and then through a method that they could explain better than I can, they do a reciprocal blast protocol where they take the blast hits and blast them back against the database and iteratively build up the catalog of elements that are present in these various primates.
Sort of a mother of all blasts approach.
Exactly. Iterative blast.
Iterative, yes.
And so we end up with a catalog that has hundreds of HERV-T elements in. Most of them are what we call solo LTRs, so solo long terminal repeats. So the repeat sequences at the end of a retroviral provirus, they have a tendency to recombine with each other, deleting the intervening sequence. So often 90% or more of the elements you find are solo LTRs. They are quite useful for understanding the evolutionary history of a retrovirus, not very useful if you want to rebuild an envelope gene. So we catalogued all of these proviruses, chose a subset of near full-length proviruses to rebuild a phylogenetic tree, found that HERV-T exists in 3 separate lineages in the genomes of modern primates. One of those lineages, interestingly enough, looks like it's been propagated purely as a passenger, because it's a set of proviruses that look like they've been shredded by the apobec proteins, the cytidine deaminases that cause hypermutation of retroviruses, lethal hypermutation. There's a whole bunch of HERV-T proviruses that have nearly exactly the same pattern of hypermutation in the genomes of modern primates.
I like the word you use, shredded. That's great.
Yeah. Does that fits with your
love it.
Your pedants? Or?
No, I think making it alive, we're using those kind of words is fine as long as they're not misleading.
It's not literally shredded.
Yeah, of course.
Right.
Yeah. So having built this catalog of HERV-T elements, then you can start to try and deduce what the ancestral sequence is. And there are programs, algorithms for doing that. And you end up, interestingly enough, starting with a set of proviruses that don't have complete open reading frames. They're full of stop codons, frame shifts, and so on. But when you do the alignment and the ancestral reconstruction, magically what comes out of that analysis is a provirus that looks like it's intact. So it has LTRs, full-length GAG, POL, the structural protein and enzyme open reading frames, and a full-length envelope open reading frame. And interestingly enough, in this case, and this was only revealed by the ancestral reconstruction, there's an additional open reading frame towards the 5' end of the genome. We still don't have any idea what it does, but it's a transmembrane protein. Looks a little bit like HIV-1 VPU in its organization, but if it did what VPU did, then we'd have had a different paper. We still don't have any idea what it does. But nevertheless, its existence is revealed by this sort of ancestral reconstruction.
So in this phylogenetic tree that you present of HERV-T3, in the lineages that wind up in humans and two of the ones that wind up in macaques, on the tree, you've got little vertical lines at the end of it. What does that mean?
So those mean orthologous loci. What that means is that is probably from a single provirus that integrated into the genome of the common ancestor of macaques and humans.
Right.
I see.
Okay? So when you're doing your ancestral reconstruction, you sort of weight these a little bit. You don't simply start with the modern, all the modern proviruses. You go reconstruct the ancestor of that little sub portion of the tree and then feed that into your ancestral reconstruction. So these proviruses are of varying ages. So the youngest is about 8 million years old. The oldest is about 30 million years old. And so, as you can imagine, they are differently present in different ranges of modern primate species. So that's
now, this mystery open reading frame is obviously kind of eye-catching. Do we know that you got the whole thing? I mean, that there aren't other mystery open reading frames that are missing from this, that just because of the nature of the search algorithm you wouldn't have found?
We don't know that. There is the sort of inescapable fact that if you didn't find it, you'll never know whether it was there or not.
Because you were looking for Gag, Paul, and Env, and then you also found this other thing that happened to be nearby.
Yeah. So one thing we do know is that this virus its closest modern retroviruses are the gamma retroviruses. So murine leukemia virus, feline leukemia virus, that group of viruses. In all cases, modern cases of gamma retroviruses, they don't have additional open reading frames. They have simply Gag, Pol, and Env. However, some of them do have what's called glycogag. That is an extra coding sequence, 5' to the GAG gene, that actually is expressed fused to GAG to make a protein that actually antagonizes an antiviral protein that I think you must have discussst on TWiV, Serinc5.
Yes, absolutely. We had Jeremy Luban on to talk.
So in modern gamma retroviruses, this an N-terminal extension of GAG provides this antagonist. We have, of course, looked to see whether this 5' open reading frame is a Serinc antagonist, and it's not, at least not in our hands. The problem with that, of course, is you have to put a little bit of faith in the fact that you've accurately rebuilt your ancestral sequence.
Yes.
Things can go wrong. The fossil record is not complete, and it could be you get one amino acid wrong and you inactivate the function of that protein. So we've done a couple of these where we've rebuilt ancestral sequences and the protein just doesn't work, right? And troubleshooting that, as you can imagine, is difficult.
You're drawing a picture of a dinosaur and you may have forgotten the feathers. Yeah,
exactly. Exactly.
That's great.
And then if you try and make it fly, it doesn't.
It's not going to work.
Yeah.
Right?
So these circulated from 30 million years ago to 8 million years, roughly. Right?
Well, this family is actually probably a little older than that. There are some elements in New World primates that would put them older than 35 million years ago. We rebuilt the common ancestor of what's present in modern, one lineage that's present in Old World primates. So it's a bit younger than the oldest element in this family.
So just a general idea or thought. So these infected our ancestors at some point, right? And obviously didn't kill them, or as you would say, didn't have a big fitness cost because the lineages are still here today, right?
Well, you would think that, but look at what's happening to Australian koalas right now. Right. So,
do you think they're going to be wiped out?
I don't know. I don't know enough about. Obviously, the larger their population, the better chance they have of a few individuals acquiring resistance. And actually, one way they could acquire resistance is through mechanisms similar to what's described in this paper. So
this could have caused a genetic bottleneck for some early hominid, but we won't know.
Right. And while we say in the paper that this replicated over 25 million years, we have really no way of knowing whether that was episodic.
Sure.
Periods of lots of replication interspersed with periods of no replication or more constant over that time. You can get some information by sequence gazing, but we don't know exactly.
I mean, one thing that I always think about is that right now humans, we have 2 known human retroviruses, right? Which we can intervene to the extent that we survive. They're not going to wipe us out. There's nothing else as far as we know, but there must have been a time when there were others and they're gone.
Undoubtedly.
And in thousands of years, if we're still around here, we're not going to know anything about HIV and HTLV because they don't integrate, right?
Well, they do integrate. They don't integrate
in the germline. In the germline, yeah. So,
so probably most retroviruses never become endogenous.
Yeah, okay.
Or most lineages of retroviruses.
Right, and since all the papers on them are digital, we won't have any memory of those either.
Although it does have to be said that the representatives of the lineages, all modern genera, modern retroviruses, you'll find things that are related to those.
Right.
In most cases.
So let me end this with this. At some point, there may emerge a retrovirus that does integrate into our germline. We don't have it at the moment, but given our past, why not? Right?
We think we don't have it at the moment, but HERV-K has been presumably inactivated sufficiently recently that we can't be 100% sure that there isn't a small population of people somewhere in the world where HERV-K is still replicating. It's also possible that the right series of recombination events could regenerate HERV-K. Actually, Thierry Heidmann sort of did that experiment. He could, taking 3 proviruses that are present in modern human DNA, you can stitch them together in such a way to make a virus that can do a replication cycle.
Okay. All right, let's get back to her.
Okay.
I'm sorry, is Kathy there? I want to make sure you didn't drop off.
I'm still here.
Because, sometimes it disconnects and then...
I'm here.
And you're frantically texting and I'm not paying attention. Okay.
So anyway, armed with these sequences, we deduced the ancestral sequence of the envelope protein. We synthetically built that envelope protein and we put it on the surface of murine leukemia virus particles that carry a reporter gene, and you can measure the titer of those viruses in the same way you'd measure any other retrovirus. And the envelope protein works extremely well, works as well as any modern gamma retrovirus, about 1 million infectious units per mil. It infects most mammalian cell lines.
You don't do plaque assays for those, right?
No. So, for the most part, you can do plaque assays for retroviruses. We find it much more convenient to do flow cytometric assays. You can do things like it's much easier to derive 2-hit kinetics and those sorts of things, wouldn't you? Have higher multiplicities. So yes, these viral pseudotypes, as we call them, infect nearly all mammalian cell lines except some rodents, and they also don't infect avian cell lines, at least this chicken cell line that we analyzed in this paper. And so when you have a situation like that, a highly infectious retrovirus that does not infect certain species, that's just what you need to go receptor hunting. Okay, so that's indeed what we did. We made a cDNA library from human cells, from the particular human cell line that was the most susceptible, made it in an HIV-based vector, and then used that vector to introduce the cDNA library. And for your non-specialist listeners, a cDNA library simply means a collection of DNA molecules representing all the genes, or as many of the genes as you possibly can, of the cell from which you derived that cDNA library. It's a way of functionally testing all the genes that are present in a particular cell. So we put that cDNA library into chicken cells, which normally are very poorly susceptible, and then we challenged those chicken cells with murine leukemia virus, with our reconstituted HERV-T envelope on the outside, and an antibiotic resistance gene on the inside. Okay, infect the chicken cells, and then use the antibiotic to select those cells. And so cells that had acquired the receptor from the human cDNA library should have a better chance of surviving that antibiotic selection because of the antibiotic resistance gene we introduced. And so you go through a couple of iterative cycles like that, and we found we got some chicken cells that had become susceptible to infection. You go into those chicken cells and you use the HIV vector sequence that you used to introduce the cDNA to fish out the cDNA for the receptor gene.
Fish out.
Yes.
You think, Dickson?
I'm thinking. Good thing you didn't do it in Drosophila. It would have been fly fishing.
So if you asked a class today how to identify a receptor for this ancient envelope, they would probably say CRISPR. Takes human cells and
it's the answer to everything.
Knock out,
right,
and then look for resistant cells.
Right.
Is it something you thought about, or?
We did. At the time we were embarking on this project, CRISPR tools were just becoming available.
Okay.
In this case, there's a reason not to go that route, though, because typically gamma retroviruses use transporter molecules as their receptors. The great disadvantage of those CRISPR screens is that if the gene that you're after is essential for the viability of the cell, you're just not going to get it.
Right.
The cell has to be able to survive without those.
Plus, it seems to me that negative selection might come up with a lot of stuff other than just the receptor.
Yes.
Good. So I recently did a session with a virology class where we did our original polio receptor cloning, which is similar to this, except we introduced genomic DNA. Right. And then the norovirus receptor paper, which was just published, where they did use CRISPR,
right,
just to contrast these two ways. And they do get other genes, but those are interesting too. But, the top hit was the receptor in that case. So.
Yeah. There are many, many ways to go receptor hunting.
Sure.
And each lab has their own bias. One method isn't better than the other.
Sure.
There are many ways that all of these methods sometimes work, sometimes don't work. This particular method has worked repeatedly for gamma retrovirus receptors. Actually, it turned out that we were very, very fortunate. After this screening procedure, we isolated single cell clones and went in and tried to get the receptor genes out, and we got several different clones. It looked like they were all derived from a single cell that had been transduced with the library. And I won't go through all the calculations we made to try and make the screen more easy. So when you're doing a screen like this, you'd like to get one human gene in each chicken cell. Yeah. So we did all those calculations and did the screen a number of times, never worked. And then in sort of desperation, we said, well, let's just bung as much library onto as many cells as possible and hit it with as much virus as we can.
Sounds like a David Kempism. I don't know if you know David Kemp, but he's an Australian molecular biologist, and he says if you don't get it to work the first time, just bung in another microliter. And indeed, things worked. You bung in another microliter, and son of a gun, I got a result.
Pretty much what we did.
So what you're saying is you shouldn't overthink things sometimes.
Right. Well, think about it first, and if that doesn't work, then
that's right.
Bung some more in.
Get a bigger hammer.
We were actually quite fortunate to identify a receptor gene by this method, and it turns out to be a transporter molecule. It's called MCT1, monocarboxylate transporter 1. It has a known function. It moves pyruvate and lactate in and out of cells, and as I mentioned,
it's important.
So it's interesting. Would the cells have survived if we had done the CRISPR screen? I don't know. I don't know.
You could do it now.
Yeah.
Well, you have cells that apparently don't have this thing. Is that correct? Or the cells that are resistant? Yeah, they don't. Are they lacking this gene?
No. So the chicken cells do indeed have this gene. It's just different enough in sequence. We think that the
mouse cells as well, right?
Right.
So do you know if anybody's ever made a knockout mouse or something like that lacks this gene?
I don't know that, actually. We might come to the end as to why the actual identity of the receptor might be interesting for things going forward. So anyway, we now were able to identify this receptor. As I told you, it's a transporter molecule. I think I also mentioned that essentially all of the proviruses that are present in the genomes of modern organisms have defective proteins. Mm-hmm. So that's almost true, but not quite true. So there's in fact a provirus in modern hominids that has what looks like an intact envelope open reading frame. Okay, so we actually realized this rather late in our studies. We had somehow missed it during that very sophisticated blast screening approach that I told you about. But nevertheless, we having found that there is indeed what looks like an intact envelope open reading frame in the human genome, we cloned out this envelope protein and asked whether it could do the same thing as the reconstructed ancestral envelope and mediate infection. And in fact, it couldn't. It's completely dead. And when we looked at its sequence, we could figure out why that was. There's clearly mutations at a site in the envelope that's normally cleaved by a cellular protease to make the envelope protein mature and fusogenic. So you'll know that envelope proteins of many viruses typically have a transmembrane domain and then an extracellular domain. The extracellular domain typically helps bind receptor. The transmembrane domain will mediate fusion. The same's true here, but that cleavage site that separates those two proteins seemed to be mutated, particularly mutated in the copy of the envelope gene that was present in humans. And so because of that, but not only because of that, there are other mutations that cause inactivation in the envelope protein. This envelope protein, while it looks intact, can't mediate infection. Okay, so then we come to the question, why is it there? Okay, and perhaps before we even get there, should we be surprised that this envelope protein is preserved?
You might think it's doing something. Right?
I'll tell you, I would be when I look at this figure that you've got here that has the human, the gorilla, and the orangutan provirus,
right?
With all the stop codons.
Yeah.
It's so blatantly obvious that the envelope gene has been preserved. That would just blow my mind if I came across that. I'd say, whoa.
Yes.
Something's going on here.
Yep.
So we approach this in two ways. One is to draw a picture in exactly the way you said and see that the envelope gene is blindingly obviously open, preserved compared to the neighboring genes. But we also did what's in theory a more rigorous statistical test. That's to take a sequence that's ancestral in hominids, the ancestor of this particular provirus, as it integrated before orangutans separated from the remaining apes, and then do an in silico evolution experiment. So simulate natural mutation and ask how often do you get an intact open reading frame at the end, and the answer is 6% of the time. Okay, so it's sort of borderline statistical significance. But as Rich said, we're much more convinced by just drawing a picture, and so this thing looks like it's been preserved. So
before you go on, let me ask Kathy if you'd like to give us your pick so you can leave.
Sure. My pick is about a teach out. So a teach-in started in the 60s. They actually started at the University of Michigan. They were ways to inform some community, pretty much from what I gather, a local community about a topic, and I think they tended to be political. And the corollary of that for the 21st century is a teach out. There's going to be a teach out. There's already been two so far. This is something new, started at the University of Michigan. The one coming up, May 5th to 7th, is Stand Up for Science, Practical Approaches to Discussing Science That Matters. And what it is from Friday to Sunday. It's sort of like a MOOC. It's being hosted by edX, and you can go, you can participate actively, you can just sort of sit on the internet sidelines, as it were, and it's going to have things about benefits and challenges in science communication, how to practice your science communication skills, some opportunities for science communication and public engagement near you. And this is being sponsored by a group here called Relate that runs summer workshops and shorter one-off kind of workshops about science communication, particularly to the non-expert audience. So this will take you to the link where you register for this, and you register with your name, and then there's a place where you can put in what will be your screen name or your anonymous name. So if you want to participate anonymously, you can. And the first two evidently have been very successful. They were on other sorts of political topics, but this one is just a nice follow-up to the March for Science. And for people who want to figure out ways that they can do more science communication, check this out.
This is great.
So is this the same edX that does the open courses at MIT?
Yes. That's okay. Who hosts Michigan's MOOCs.
Got it.
So that's why it's edX.
Okay.
Yep.
The co-founder, I see that the co-founder of Relate, Elise Auerbach, was the former. This is great. The former co-boss of Nerd Night Ann Arbor.
Right. So part of their summer workshop is that you go for, I think, an hour and a half or 2.5 hours once a week, and you start crafting a way to talk about your science to the non-expert audience. And the graduation, as it were, is you go to Nerd Night or some other event that's up at Michigan State and give your talk in a bar.
Great.
Cool.
Terrific.
Yeah. So Relate was started by 2 graduate students who are now postdocs, and they're trying to make sure that there's sustainability throughout the university. But meanwhile, they've been called on to participate in this teach out.
Cool. Thanks,
Great.
Kathy. So now you're off to do a concert. Is that right?
Yes. Singing with Choral Union and the Toledo Symphony in Toledo, Beethoven's Missa Solemnis.
Enjoy. Have fun.
Excellent.
Fun.
Thanks.
All right.
Okay.
See you next week.
See you.
Yep.
Okay. Bye-bye.
Bye.
Bye-bye.
So we left off with the finding that this is unusual that there's an open reading frame in this envelope. Right.
So this particular provirus has been in hominids. It integrated between the time that gibbons diverged from the remaining apes and orangutans diverged. So it's present in orangutans, humans, and gorillas. Not in chimpanzees, actually, because chimpanzees have had a segmental deletion. Segmental deletion of that locus.
And
so
bonobos?
And bonobos?
Bonobos, I don't recall, actually. We may not have even looked in bonobos. Okay. But what isn't there is an empty pre-integration site. That evolutionary theory would be in trouble if there were. Okay.
So
at least what we think about the common ancestry of...
so it was there and it got deleted, right?
Right.
Okay.
Right. The whole locus got deleted. So this thing has been in the hominid genome for between 13 and 19 million years. Okay? And has been preserved through that time.
Is a protein made?
The protein, we don't know, actually. So the RNA is present in thyroid and at low levels in other tissue, but really in high levels only in thyroid. We've not been able to find a cell line that expresses it, which obviously would have been helpful for these studies. But so we don't know if it's made in modern humans.
Got it.
Okay. So what we next did was to take the modern human protein, the sort of 13 to 19 million year old ancestor of the modern protein, which also is non-functional, by the way. And then the 32 million year old ancestrally reconstructed protein that is functional. And what we then do is to express those in cells and then ask if that affects their susceptibility to infection by the HERV-T pseudotype virus.
The one that you reconstructed, right? Yep.
Yeah. And the answer
is... So you're theorizing that this preserved ENV may be some kind of an antiviral.
Right, exactly.
May have been accepted.
And we had a reason to think that because receptor interference is a well-known property of retroviruses. There are examples of laboratory animals, mice and chickens that have resistance to retroviruses because of envelope proteins in their DNA, endogenous retroviral envelope proteins in their DNA. And so what we could then do is by expressing these proteins either in human cells or in chicken cells where we'd put the cloned receptor in, we could show very clearly that all of those proteins quite efficiently block infection. They block infection specifically by the HERV-T pseudotyped retrovirus. They don't block infection by murine leukemia virus with its own envelope protein. And so finally, what we could then show is that expression of these envelope proteins actually cause removal of the receptor from the cell surface. So it's actually a global reduction in the amount of the MCT1 protein that's made when the retroviral envelope protein is made, and virtually none of it's present on the surface. So it's basically removing access to the receptor. That is what this endogenous retroviral envelope protein is at least capable of doing.
Pulling in the welcome mat.
Right.
Which has been shown for other envelopes, right?
Yes.
And I always wondered why other viruses didn't do the same thing. So there's no example of.
Well, they don't actually have conscious thought about that.
Yeah, I know. But
I mean, viruses other than retrovirus.
Yeah, why hasn't that?
So this virus here on the table, poliovirus, I guess you can't make a single protein to do the same thing, to downregulate the receptor. You need the whole capsid to bind the receptor, so that may explain that. But other viruses have glycoproteins that could individually be, they don't integrate. Maybe that's part of the thing, right?
Yes.
Although we know that some viruses, there's accidental integration, like these Borna in 13 striped ground squirrels, where one protein is apparently protecting. The DNA has integrated, and it's protecting animals from infection. So maybe there are other precedents. You know about that story?
I don't know about that story, but it is nonetheless true. There is in principle no particular reason why receptor interference shouldn't be active in other viral systems. Obviously, the ability to integrate into the host DNA puts retroviruses.
So these capsid proteins that are being made as an interference for the virus attaching envelope, is that actually on the surface of the cell then after it's made?
No, it seems to drag the receptor off the surface. So likely what happens is at some point in the secretory pathway, the retroviral envelope protein and the receptor protein meet up, and there's some fate alteration for the receptor. It seems to be degraded. We've done a few little experiments to try and figure out how that degradation occurs. We're not really ready to say anything definitive about that yet. But that's an interesting question, actually. How do these envelope proteins cause removal of the receptor?
Right.
So give us this scenario now of what you think happened.
So overall, sort of summarizing, what it looks like happened is that somewhere between 40 and 30 million years ago, HERV-T began to use MCT1 as a receptor. Okay, presumably it was derived from some other retroviral lineage that's been lost in the sands.
This is some primate ancestor, some Homo sapiens ancestor, right?
Right. Well, it has
way
to be
before
ancestor
that,
of all
the hominid
modern
ancestors.
primates.
Yeah.
Right.
So because this virus is also in New World primates. Okay. So it's older than that split.
Right.
Okay. So at least in the old world primate lineage, the HERV-T replicated either continuously or episodically for the next about 25 million years. Okay.
Obviously didn't wipe out everyone, otherwise it would be gone. But okay.
There would be no TWiV.
Right. That's right.
Or even outhouse.
So somewhere between 13 and 19 million years ago in the hominid lineage, okay, so orangutans, humans, not before that, this provirus that to this day maintains an intact envelope protein, that protein was, that gene was incorporated into the modern hominid genome.
But probably by infection, right?
Yes, almost certainly by infection.
By a HERV-T like virus?
Yes. And then HERV-T presumably continued to replicate, but the last the last time it integrated into the germline and left a fossil or something like a fossil that persists to this day is around 11 million years ago. Okay. 11, so if you think of the timing, sort of 30 to 40 million years ago, and then replication, and then about 13.
Is that like Lucy?
Era? No, it's much earlier than Lucy.
Earlier than
Lucy. Yes.
Okay, all right.
Lucy's 2 or 3 million, I think.
Okay, okay.
This virus was long gone before.
All right.
So in fact, calling it human endogenous retrovirus T is a bit of a misnomer. We only call them human endogenous retroviruses because they're in the human genome.
Yeah.
Most of these endogenous retroviruses are not specific to humans.
When you remove the receptor, I mean, that's a functional molecule for the cell, right?
Yeah.
Right.
So what is the consequence of that? It's like doing a knockout on yourself.
Right. So we can make stable cell lines expressing the envelope and sufficient levels of the envelope to cause resistance to viral infection. So either the cell can survive without the transporter, or with very low levels of it,
or with
the receptor, or with the envelope bound to it.
God.
But that's cells, right?
Yeah. What we do find, however, is if you don't clone those cells rather quickly, then the envelope expressing cells tend to get purged from the population.
Right.
So it's toxic in a small way.
I hear you.
And you said in humans the RNA level of this is very, very low.
In most tissues,
so maybe it's been downregulated for that reason.
Yeah.
We don't need it anymore, right? Because we don't have any.
Right.
Yeah, but in the day when we did need it, it would still metabolic consequences.
Yeah, so either you get infected and get a serious infection, or you have a little
energy
issue.
Energy issue.
And
so we say that we don't need it anymore, but there is a
the envelope. You mean the envelope, right?
The envelope. We don't need it anymore, but there is a little wrinkle to this, and it's in the interpretation of these findings. So we've sort of presented the story that this thing evolved and is preserved in order to provide defense against this virus, and that's a plausible, likely scenario. We don't know for sure that that's true.
Sure.
Right. There's another possibility, and that what this envelope protein does is actually regulate the process of pyruvate and lactate transport. Okay?
Right.
Sure.
Yeah.
That's right.
And there's a reason. There's actually a very good reason why that might be important. It turns out that this MCT1 protein is dramatically upregulated in a subset of cancers. Okay? You know that some cancers really like to metabolize using glycolysis. And what they do is they dramatically upregulate this receptor.
The Warburg effect.
Yes, exactly. So you could imagine having something like this to counteract that might be quite important. And that actually brings us to a project that we've just started. Knowing that MCT1 is dramatically upregulated on a subset of tumors, what we're now going to try and do is use this envelope protein to make oncolytic viruses.
Hey,
that's brilliant!
Nice.
So it's not likely that this envelope is upregulated in those tumors, right?
It's possible. We haven't yet looked. That's actually well worth looking.
Right.
But
is this through a gene duplication that it makes more MCT, or how does it make more?
That I don't know, and I'm not even sure that it's known. My thought is that it's transcriptional, but it could be through amplification.
Okay.
Yeah, that's cool. That's a neat idea.
Yeah, but nevertheless, I think if we can generate an envelope protein with the right affinity that will selectively infect cells with high levels of the receptor, then
wow.
So we're going to
try that.
So the idea is the oncolytic virus will be downregulating MCT1, which you think might be essential for the tumor growth?
No, what we would like to do is use a really cytolytic virus, like VSV, for example. Give it the
I see.
The HERV-T envelope protein, and so that it selectively replicates
MCT1,
and kills the tumor cells.
Right.
But it could also be that downregulating MCT might have an effect on the tumor.
That would be probably more easily accomplished with drugs. Though,
drugs,
I think.
That's cool. You're just starting that. Wow, that'll be neat. Hope it works.
Me too.
It could be another TWiV paper, right?
Yeah.
That's a great story.
That'll take a while, I would think.
Yeah, that's a great story. I think that this is a great example of why mining these endogenous retroviruses, you never know what's going to come out of it.
We couldn't possibly have anticipated that this... and this sort of work really doesn't get funded by NIH. It's just a little bit.
It's a fishing.
So that's why having Howard Hughes money, you can use it for this, right? And they don't mind you doing things as long as you get nice papers out of them, right?
Right.
Even if you prove to them that you're a very good fisherman.
I'm actually not a terrible fisherman.
Really, you do freshwater things?
I fished a lot as a child, and I've just recently started picking it up again.
Oh, cool.
I went striper fishing with Steve Hughes a couple of weeks ago, and I've been taking my kids to Harlem Mere to pull out bluegills, and there's even one or two bass in there.
Look at you.
Wow.
Yeah,
it's fun.
All right.
It is fun. You're right.
Should we do some picks, gentlemen?
Sure.
We no, I'll give you some advance notice, but we pick something you might want to. If you,
I've got.
Well, you heard.
I want to
Kathy's.
sleep a little bit.
I've got something.
I think.
By the way, Kathy texted to say she wanted to mention that this teach out is free, and looking at the website, it's free, or you can pay $25 and they'll give you a certificate when you finish it.
Really?
I think they should give you a TWiV mug or T-shirt, but well, in exchange for your coming here, we got you a TWiV mug there.
Yes,
so enjoy it. That's a special edition. You can't buy it. We had them specially made for guests. And
wow, that's wonderful.
It's cool.
Thank you very much.
You're welcome.
I shall treasure it.
Dickson, do you have a pick for us?
I actually do. Actually, first got interested in Saturn when Carolyn Porco came on a Nova show. She came on a Nova show and just explained what they wanted to do. And then, of course, they did it. And there was some trepidation about it because the Cassini satellite contains a nuclear reactor that actually generates energy for it. Mm-hmm. So there was a fear that it might blow up in the atmosphere before it actually escaped into space.
I remember that.
But nonetheless, it did, and it was very successful.
It did escape without blowing up.
It did, yes. And it reached its destination and took some fabulous photographs of this amazing planet. And then I had the thrill of meeting her at a TED Talk. She was giving a TED Talk at the same one that I was at, and she was great, and she spent lots of time, and I had a chance to ask all my dumb questions about outer space, and and she was, I'm sure, at one point looking around. Oh, gee, I wonder where the Coke machine is or something. But nonetheless, she was very accommodating, and she appreciated my enthusiasm for just an interest in NASA projects. And then today, we find that we're now in the end of the tether, so to speak. The Cassini mission is over. How could you say it's over? I think they ran out of money because otherwise it should still be active. I don't know why they're doing this, but they're going to plunge the satellite into the center of Saturn. But before they do, they're going to send it as close as they can to the surface of Saturn. And yesterday, they passed between the rings and the planet itself, and they took a picture. And I can't interpret this picture. It looks like a whirlwind of some sort with a center in it, and it doesn't make any sense. But they're going to actually try to get pictures of the ring particles and to see how big those snowballs are and stuff like that. I'm absolutely thrilled at this. And it's really too bad that it's ending, but such a legacy of success. I think NASA should be incredibly proud of what they've done. And, of course, I've named my pick Lord of the Rings, but we want to use that for something else.
So you once picked her as a pick, something about her.
I did, because she's a remarkable communicator of science to the public.
Cool.
She's fabulous. She's so enthusiastic, and she's so energetic, and she's so literate. And she doesn't use big words. She just tells you what the thrill was to see those first pictures from Cassini come back, that you felt as though you were in the same room with her.
Astronomy is particularly interested to a lot of people, right?
It is. And by the way, it attracts a lot of women to it because I think nobody wants to stay up that late.
So could they have kept this the Cassini orbiting?
I think so.
Forever?
I think they could have. I really do think they could have. I don't think there was they're directing this thing to go between the rings and the planet. That wasn't by accident.
I think it reached a point in its life cycle where these things do break eventually, and they've gathered a whole lot of data about Saturn and done a whole lot of projects with it, and they wanted to do some stuff that would require a one-time plunge, and so
sure,
it's the next step in the process.
Isn't there something still traveling out?
Oh,
Voyager, but we don't hear from it anymore.
Gone. Yes, we do.
We do.
Yes. So the signals are still coming back. Yeah, very, very slowly.
Yeah. How far out is Voyager?
It's almost at the edge of what we would describe as the gravity pull from the solar system.
So can it tell us whether Pluto's a planet?
No, but Neil deGrasse Tyson can. He doesn't think it's a planet.
Thank you, Dickson.
Dickson, these pictures are amazing.
Yeah, they are fabulous.
Alan, what do you have?
I have a video that I mentioned, or a video channel on YouTube that I mentioned when we were in D.C. This is the channel for the Lawrence Livermore National Laboratory, and specifically this is a playlist of some films that they are just now declassifying. These are atmospheric nuclear tests that were done from 1945 to 62, before the Comprehensive Test Ban Treaty went into place. And these were classified for many, many years, but now they've reached the point where they can they're reviewing them, and they've got an opening video talking to a physicist who's working on this project, who's working with somebody from Hollywood who does film preservation, and a lot of these old films are in the process of falling apart, so they wanted to digitize them.
Right.
And you can go and look at all these different detonations, atmospheric detonations, from the different angles that they shot. They're sorted by which operation it was, and they describe what type of bomb was going off. But they're all nukes, and they're all going off in classic mushroom cloud formation from different angles, some quite dramatically.
Yeah,
this is really horrifying.
I find these haunting, and yes,
they are. These are. If you like mushroom clouds, there's plenty of them here. It's amazing that they did so many atmospheric tests. This is between 1945 and 62. I was telling my wife yesterday, when I was a kid, in the early 60s, they told us when it rained, not to let the rain go in your mouth because it was radioactive from all these atmospheric tests.
Well, in fact, you can date
you don't believe that?
They were wrong.
No, you can date tooth enamel of bodies or just teeth that have been pulled.
Mm-hmm.
Yeah, based
on for people who were born during the time period that this fallout was circulating, which actually includes me. So for several years after the tests ended, there was still this radioactive pulse in there from all these tests. It forms a distinct geological layer. People have proposed using that as the marker for the Anthropocene era. Because a million years from now, some future alien civilization will be able to take a core sample on Earth, and they'll say, hey, wow, this is weird. There's this pulse of radioactive material right at this point. So it got into everything, and this was a big part of what we did with our defense budget.
Yeah, you could imagine millions of years from now, as we wonder what killed the dinosaurs, people are thinking what killed the humans. They find this layer.
That's right.
They'll have their answer, irrespectively of how we...
Yes.
By the way, these were shot at 2,400 frames per second.
Yes, they used state-of-the-art film cameras of the time, which are actually still pretty impressive.
Just for reference, when I shoot video, it's about 30 frames per second.
Right.
Right.
So these are mostly in slow motion.
Yeah.
They must have some big hard disks to store them all on there. Right?
I don't know if they digitize them at the same rate, but yeah, it does take up a lot. Rich Condit, you have for us.
Man, after that, I just want to go take a nap or something.
It's what we do, Rich. It's what we do.
Okay. On a lighter note, this arises from a conversation I had in February when we went to Legoland with my son, who's the guy who works at Google. And he gave me an article that's a New York Times Magazine article that's entitled The Great AI Awakening, AI being artificial intelligence. And it basically describes a paradigm shift, at least in practice. I think in theory it's been going on for a while, a paradigm shift in machine learning, artificial intelligence. And I will try and describe it. In the old model, if you wanted to have a computer, train a computer to recognize a cat, you would give the computer all sorts of rules about pointy ears and whiskers and fur and a tail and four legs and that kind of stuff, and try and train the computer to recognize a cat that way. The new model uses what the computer scientists call a neural network. That's not hardware, it's software that basically consists of a bunch of nodes that, for us behave kind of like neurons that have connections with a bunch of other nodes and actually exist in layers. Well, you'll have a layer that's a bunch of interconnected nodes, and they interconnect with a higher layer that does the simplest level of task, and then there's a higher, more complex layer over that, and ever more complex layers over that. And this neural network, what you do rather than trying to train it with the rules is you just show it thousands of pictures of cats. And it figures out what a cat looks like. And in fact, you don't even know how the computer did it. You can't go back inside and say, how did you do this? But it does it, and it does it better than the old model. The article makes this point. Same process was used in Google Translate. There was a order of magnitude shift in how good Google Translate was when they applied this model. And I've done my own tests recently with some of this stuff, and Google Translate actually can be pretty good. At any rate, I thought that was a fascinating concept, so I give you that article. This is sort of a nested series of picks because in the process of looking at that article, I had to look up what an artificial neural network was, and I give a Wikipedia link to that. But then what came up that my son pointed out to me is this thing called Deep Dream. And let me give you his description of it. Sorry, I got to find it here because, here it is. Because this is really pretty good. He describes it better than I could.
Deep night.
Someone at Google playing with one of these programs decided to basically run it in reverse. Basically amplify portions of the image that triggered high-level neurons. The result is these trippy/disturbing images where eyes and faces emerge from the background of the image because those are the high-level features that the neural network is trained to respond to. And I've given a link there to Deep Dream images, and they are spooky.
They're disturbing. Disturbia. Welcome to Disturbia.
Quite.
So at any rate, yeah, I mean, it really is like I can see why they call it Deep Dream.
Boy.
By the way, the section of Google where they do this stuff is called Google Brain. I just thought it was fascinating. One of the things that fascinates me is that you set a computer up to do this stuff, and the computer does it, and you don't really know how it did it. I asked Jer if you could, maybe reverse engineer it, figure out how it did it, and learn something maybe about how the brain works. And Jeremy's response was, I suspect you could try and reverse engineer a trained neural network, but that it would be difficult in practice to assign any human meaningful concepts to individual neurons. At any rate.
That's amazing. And I particularly am amazed by the translation part. I mean, and now it's translating really, really well.
Yes, right. And it's the same thing. You use a neural network and you give it lots of examples of how something should be translated. And the computer essentially makes up its own rules.
Incredible.
And it's exactly the way it happens with humans, right? You're born with a brain,
sure,
and you encounter examples and you figure it out.
Amazing.
And some of us continue to make up the rules.
and what is coming down the pike?
Who knows? Who knows?
It's going to be something.
Thanks, Rich.
Yeah.
Paul, what do you have?
So I thought I would pick the website of the Howard Hughes Medical Institute.
Mm-hmm.
Now, to us scientists, we're all familiar with the way that Howard Hughes Medical Institute funds scientists to do their research, and you can find out all about that on the HHMI website. But what is perhaps not as well known is that HHMI has a huge effort in science education.
Mm-hmm.
And on this website, you can find all sorts of resources, from teaching guides to videos to essentially anything you might imagine. Hundreds and hundreds of different pieces of science information, something that a science geek, someone who's really interested in science, could spend days, months immersing themselves in this material. So, and it's all made to be accessible to the general public and for school children. So
we might have one or two listeners who'd be interested in something like that.
You bet.
Yeah. I'd really encourage people to take a look at it.
Cool.
HHMI spends quite a lot of resources on education as well as scientific research.
Yeah, I picked Tangled Bank Studios once, which is the
did you? Yes,
the film studio they fund,
right?
Which is really good. I have tried to get them to fund TWiV, but I emailed Sean Carroll, who's the education head, and he said, "Well, it's really nice what you're doing, but we want to do our own thing," which is too bad because we don't need a lot of money, do we?
Apparently not.
Well, yes, we could.
If we had a lot of money, we could spend it.
We could spend it.
We don't even need a lot.
We could probably use a soundproofing on your room here, Vincent.
Yes.
Well, if I run into him, I'll lobby on your behalf.
Would you please?
That would be nice.
I can't promise anything.
Yeah, of course. Well, it's probably. No, he writes nice science himself. One of my picks recently was a book, Endless Forms. I forgot the rest of it, but it's based on Darwin's quote. But it's all he explains development really beautifully because he's spent a lot of time in his career studying development of. I think he studies worms or flies. I can't remember what, but in Hox genes, and he did a really good job in his book. So that's great. All right, I have two picks. One of them is a rewriting. Speaking of Google rewrites its powerful search rankings to bury fake news. And so they're not going to get rid of it because they're trying to purge it from the top, all the fake news that's out there.
That's a start.
It's a start. And so they have reorganized their algorithms to do that. So I have a Bloomberg article that describes that. I think that's really good. To start at least, because that's a big problem when people look for vaccine information. They get the crap on top, and we'd like to remove that.
Yes.
And then the second pick is a video by Senator Sheldon Whitehouse. Wouldn't it be nice if he got elected president?
President Whitehouse.
Yes.
We'd have the White House on the politics of climate change. It's a 5-minute video where he explains why we have had such a reversal in climate change. It starts with a Supreme Court decision, which he says driven by the Republican-appointed members of the Supreme Court who decided that they're not going to. And he said before that there were numerous prominent Republicans who, believed what was going on, and now they've all changed.
Accepted the truth of what was going on.
Accepted the truth, and he explains it in 5 minutes. And he says whenever he goes to Congress, he has this sign that is now getting dog-eared about climate change that he talks about it. So I recommend looking at it. It's 5 minutes, and he's very convincing. And
I actually watched this video just yesterday, in fact, and tweeted it along with the caption, what's his name again? Sheldon
Whitehouse.
Sheldon for president. Yeah. I thought it was remarkable. It should be compulsory viewing.
It's very good.
Anyone interested in
it's scarier than the bomb testing.
Yeah,
it is. At least the bomb testing's over, right?
Yes, for now.
Really, we're going to be testing.
I think bomb testing is not over. If you ask the leader of North Korea.
But for us, I'm talking about. We can't control it.
No, we do it underground now.
Do we still test nuclear weapons? Really?
I don't think so. No.
I don't think we're doing it underground either.
We don't have to because we already know how they work.
Yeah, we've made them, right? They're pretty powerful. You don't need to make them more
Fairly
powerful.
powerful.
All right. We have a listener pick from Anthony. He says this is a sub-pick of Alan Dove's pick, which your pick was a Raspberry
Pi Model 3 a while ago, yes. And this is a weather monitor kit that you can attach to your Pi 3 and set up your own home weather stations. A very TWiV appropriate.
It's TWiV, totally.
TWiV appropriate.
Anthony gets it for sure. Weather monitor kit, yeah. And so that you could see if we're telling the truth about the weather, right?
Right, or at least locally. It's got a bunch of different sensors that you can plug in.
Yep.
I mean, we haven't gone into wind speed and barometric pressure and stuff like that. So
Right.
I mean,
sometimes people do. Sometimes
I know.
Sometimes.
I know, but we don't. We
don't.
No.
We
get into dew point's a big deal.
We have done that.
I've covered wind speeds occasionally. I think.
I like what Stefano Bertucci said. Is it Bertucci or Bertuzzi? I got to get this right.
You should know this.
Yeah.
B-E-R-T.
I'm disappointed.
Oh, I made a mistake last week. Stefano Bertuzzi. Two Z's.
Bertuzzi.
What he said last week, he has such a short commute that by the time he gets home, he's just getting past the weather. And he listens to TWiV. Oh, well.
Yeah, I missed the weather, didn't I?
When?
Today.
Yeah, you did.
So,
You did.
So how was it out there?
Oh, it's hot.
It's hot out, yeah.
It's hot.
Hot.
Yeah.
Very hot.
It's 28 Celsius. Or 80. And the sun is setting now, so I have a nice view of the sunset here. And we've now moved into the part of the year where it's still light when we finish TWiV, because sometimes the sun is just going down.
That's true.
That's TWiV. You can find it at Apple Podcasts, the new name for iTunes, Apple Podcasts, microbe.tv/TWiV. Send your questions and comments to TWiV@microbe.tv and consider supporting us because, the HHMI won't support us. So we have to raise money on our own. So go to microbe.tv/contribute and you can, for example, Patreon, you can give $1 a month. If everyone did that, man, we'd be all set here. We could do lots of things. But if
wouldn't be here then.
No, but
we'd have our own TWiV studio. TWiV
do what you can. It was a PayPal. Some people don't want to do Patreon, which is a monthly subscription thing. You could do PayPal, or you could use our Amazon affiliate link, or you could just buy a mug, and we get $1 from every purchase at cafepress.com/TWiV.
And the guests could have refreshments.
Exactly.
That's right.
Of their choice.
You need a glass of water? Or
It's okay. I'm fine.
We can get you one outside when we're done.
Water!
I'm sorry.
This TWiV mug is empty.
That's right. The TWiV mug
I
is
gave him a TWiV mug, but there was nothing in it.
That's the name of this episode, by the way. The TWiV mug is empty.
Should have been... do you like single malt?
I do.
What's your favorite?
Oh, everything.
Do you have a favorite?
So, Paul, there's a cabinet, I think, over to your right. You should go ahead and look through that but on the top shelf.
They're gone. They're gone. Yeah, we don't have it here anymore. Yeah, we used to have a bottle of single malt in there, and on Fridays we would. And when Alan was here, he was a student here. We used to have a drink before we went home.
Because I've thought about having this bottle of scotch in my office, but
you just do that.
I'm dubious about the appropriateness.
No, it's okay. These are ivory towers.
Stodgy old Rockefeller, come on. You still have a bar, don't you, in a faculty club?
We do.
Okay, then there you are. My case rests.
Do you go to the faculty club?
Occasionally, yes.
Yeah.
I used to know members there that kept their own bottles. And they could drink from them whenever they wished.
Really?
Yes, they did.
It's like having your own glass there behind the bar.
That's correct.
My wife was a postdoc years ago there, and on Fridays I would occasionally go down because we lived down on the east side, so I would go down there and
lovely faculty
and hang around. It was great. Yeah, it's really nice.
Yep.
Our guest today has been Paul Bieniasz from the Rockefeller University. Thank you, Paul. Appreciate it.
Very welcome. My dream did come true.
Come back.
Please come back.
I will, certainly.
Love to have you back. We love having guests and sharing the studio here. Dickson Despommier is at thelivingriver.org and parasiteswithoutborders.com. Thank you, Dickson.
You're welcome.
Hope you enjoyed it.
I had a great time.
Did you learn a little bit about fossils?
You know what? I understood most of what you guys were talking about this time. This was clear.
Good.
Clarity in science is important.
I hope other people as well understand.
No,
and you, Paul, you gave a nice summary at the end.
Exactly.
Last week we got an email from someone. He said, it would be nice if, for those of us who don't quite understand what you're saying, just give us the summary at the end. And so that was, I think you did that very well.
Yes.
Rich Condit he's a former professor from the University of Florida in Gainesville. Now he's a professor emeritus at Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. Thank you.
Of course, Kathy Spindler, who had to leave early to go sing at the University of Michigan in Ann Arbor, Michigan. Alan Dove is at alandove.com. He's also on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
Wait a minute. alandove.com. That's not right, is it?
It is.
I thought it was turbidplaque.com.
Well, you can go there, too.
It's the same?
Turbid Plaque is the science blog. alandove.com is the main. It'll direct you to either my personal site or my professional site.
Which one would you prefer us to...
just send people to alandove.com. That's fine.
All right, so we did. And I know we used to say turbid plaque, and
I think so. Yeah.
I don't know why it morphed into Alan Dove. It must be a mutating endogenous retrovirus.
Yes, that's it.
Hopefully, it will not become extinct.
I'm Vincent Racaniello. You can find me at virology.ws. The introductory music you hear on This Week in Virology is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Well, thanks, everybody.
Sure. We're going to pick a title.
We are.
Oh, yeah, title. Right.
We have a few.
We do have a few here. Paul, you got to check some of these out.
Listen, hands down, the purloined envelope.
You think hands down that's it? Like that?
There are many good ones here. Here's another clue for you all, the virus head Paul.
I like that.
I mean, that's good. And you may like some of the others, but when I saw the purloined envelope, I really liked that.
Okay, then, Paul, some of the others are really good. I like exaptation is the sincerest form of immunity. That's pretty good.
I like that too, yes. It's good.
I also like, then I saw Herv apes, now I'm a believer. Somebody likes the monkeys.
That's it, yeah.
You like the monkeys, Alan?
I don't especially like the monkeys, but for some reason I came up with that. And
So I have to admit the word purloined is a gap in my vocabulary.
Stolen.
Stolen. Okay. Stolen.
And there's a Edgar Allan Poe short story, The Purloined Letter. It was one of the first detective stories.
Exactly.
All right. So it turns out I will take lessons in English from Americans sometime.